Hi, my name is Andrew, and today I'm going to be talking about different expression metrics for RNA sequencing data. In this RDM Byte, you will learn about common units for gene or transcript expression, including some which you could argue could be retired, but are still very common in databases and publications. We'll discuss the drawbacks of some metrics and make the case for using TPMs as standard. RNA sequencing, or RNA-seq, is a high-throughput sequencing technology typically used to quantify relative abundances of RNA molecules. The majority of RNA-seq uses Illumina short-read technologies, where RNA is extracted, converted to double-stranded cDNA, and sequenced by synthesis to generate raw FASTQ files, which are then aligned to a reference genome or transcriptome. Mapped reads are then used to quantify feature expression. RNA-seq often means messenger RNA sequencing, used as a proxy for protein levels which are hard to measure directly. We can also do total RNA-seq, removing only ribosomal RNA, or microRNA-seq, a way of quantifying short, non-coding RNAs involved in cellular regulation. These methods are highly analogous, only differing in the choice of starting material. The output of standard RNA-seq pipelines is a matrix of feature abundances, typically at the gene level. Expression matrices contain expression values for each gene for each sample, and these then form the basis of all downstream analysis. Knowing how processed these values are is critically important for comparing expression within and between samples. At the most basic level, abundance depends on how many reads map to a feature, with more reads suggesting higher abundance. This is often normalized to the library size as CPM, counts per million, since the deeper the library is sequenced, the more reads per gene. However, relative abundance between genes depends on how long the feature is, as you would expect longer genes to contribute more reads to the sequencing pool. In this example, gene 3 has the fewest counts, but is short, so has a high relative abundance within the sample. Effective counts scale for gene length, taking into account that gene ends are more poorly sequenced than the middle. Effective counts are typically used in differential expression analysis by tools such as DESeq and Sleuth. When comparing between samples, it is easy to immediately draw conclusions, such as sample 2 here having more of gene 1 and 2, but the same of gene 3. But sample 2 had 4 times the reads, so actually expression of genes 1 and 2 are comparable, but gene 3 appears to have lower expression. Units make a big difference when making conclusions. As we've seen, comparing expression between samples needs to account for both the effective size and the sequencing library depth. One of the most popular metrics is FPKM, fragments per kilobase of exon per million reads mapped. FPKM largely replaced RPKM as a metric as paired-end sequencing became more common. FPKM represents sequence fragments which will be covered by 2 sequencing reads, but as the 2 reads are non-independent sequences, and will not necessarily both map successfully, we can only consider the fragment, not both reads. This is why FPKM is not double RPKM with paired-end reads, even though that is commonly seen in the literature. Whilst FPKM is scaled by library size, it is not as a proportion, so FPKMs will be inflated in larger sequencing libraries. To combat this, FPKMs are typically scaled or normalized using methods such as TMM, or by using quartile-based factorization. This is another level of processing data scientists need to be aware of. In contrast, TPMs, or transcripts per million, are a proportional metric, making them relatively stable for cross-sample comparison. TPMs can be skewed drastically, however, by large changes in high abundance genes, so plotting a TPM distribution is always a good first step. If you have a matrix of FPKMs, TPMs are easy to derive by dividing each gene's FPKM value by the sum of all FPKM values in that sample. So, to compare, all metrics mentioned account for feature depth, but the most basic metrics deviate on accounting for feature length and total library size. Only TPM is relatively comparable between samples, and also benefits from being a simple proportional value, making it relatively easy to explain relative differences. Taken together, TPM is increasingly being seen as the best metric for RNA-seq data. Other metrics are fine, as long as they are interpreted with their caveats in mind. That concludes our look at different RNA-seq expression metrics. Take a look at the links associated with this video for more information and more RDM Bytes in this area.